All right, in this unit we'll be studying the structure and function of DNA as the genetic material of heredity, and also this thing that's referred to as the dogma of biology, the central dogma of biology. And we'll come back to that later. But what we're looking at here is just to identify that these are two DNA molecules side by side, and they're models, they're drawings, they're not what DNA actually looks like, but it's so small that it's difficult to see, and so this is the way that we need to conceptualize it. We use models a lot in biology for conceptualizing things that are difficult to see. So there are two DNA molecules shown here. The structure of one molecule is a double helix, or a twisted ladder. Most often described, if you don't call it a double helix, then it's described as a twisted ladder. Double because there are two strands that are twisted around each other. That's the helix part. And like I said, DNA functions to store genetic information. It's the genetic material of heredity. So it stores genetic information, and it allows for the copying of that genetic information, so it can be passed from parents to offspring or from cell to cell. And it can also be read when it's time to make protein. And that's one of the big things that we'll be looking at in this unit, is how does all that happen? And that's all part of the central dogma of biology. We know this because of a series of 3 experiments performed by 4 scientists. In other words, we know that DNA is the genetic material. So that's all we're looking at first here, are the experiments that supported the idea that DNA is the genetic material. This first experiment was performed by a scientist named Frederick Griffith in 1928, a British scientist, who studied disease, and he was specifically studying pneumonia and the bacteria that cause pneumonia. Streptococcus pneumoniae. And he found 2 different strains of the bacteria that cause pneumonia. He found a strain that he called the S strain, S for smooth, because when the colonies are grown in cell culture, like we see here in this petri dish, the edges of the colonies are smooth. There was another strain that he discovered that had rough edges to the colonies in cell culture, and so he called those the R strain, R for rough. And so in experiment 1, he injected the disease-causing S strain into mice, and the mice died. No surprise. That's what you would expect. If you inject an animal with disease-causing bacteria, you expect that animal to come down with that disease and die. In experiment 2, he injected the R bacteria, the R strain of the Streptococcus pneumoniae, into the mice, and they lived. That was interesting, but it's not a huge surprise, knowing that the bacteria is harmless. So far, there's really no surprise. There are really no surprises here. In his third experiment, he took the normally disease-causing smooth bacteria and heat-killed them. Heat-killing simply means that they're heated to the point where the enzymes inside them are denatured. They're no longer able to function, basically, and reproduce. And that's really what makes you sick. When you have pneumonia, what's happening is that the bacteria are reproducing in your lungs, and you know, they're just having a party in your lungs and feeding off your lung tissue and so forth. So heat killing prevents them from reproducing, and therefore prevents them from making you sick. So he heat killed the S strain, which normally is the pathogenic strain. Hopefully you're familiar with that word pathogen, which means a disease-causing organism. So these are normally pathogenic, but he heat killed them, injected them into the mice, and the mice lived. No surprise, right? So far, nothing unusual is going on. But this is where things get interesting. When he took the heat-killed, normally pathogenic heat-killed bacteria, and mixed it with the normally non-pathogenic R strain of the bacteria, the mice died. That's a surprise, because these don't cause disease. The heat-killed smooth bacteria don't cause disease. They do if they're not heat-killed, but once they're heat-killed, they don't cause disease. He showed that in experiment 3. The harmless bacteria, he showed in experiment 2, don't cause disease. They're harmless. But when you mix this non-disease-causing bacteria with this non-disease-causing bacteria, you get disease-causing bacteria. And that was a huge surprise. Not only that, when he cultured the blood of the mice that died, he found live disease-causing the smooth strain of bacteria living in the mice. But he didn't put any living smooth bacteria in the mice. He put heat-killed S-strain bacteria in the mice. But he found living S-strain bacteria in the mice when he cultured their blood. So that was a surprise. So in trying to explain this, Griffith concluded that something had been transferred from the heat-killed S-strain bacteria to the living R-strain bacteria to turn them into living S-strain bacteria. But how? He speculated or inferred that the dead S-strain bacteria had released its genetic material into the environment inside the mouse, and the R-strain bacteria picked up that genetic information, which turned the R-strain bacteria into S-strain bacteria. Whatever that information was, they didn't know. So we call this today Griffith's transformation. He was the first to demonstrate that this happens in bacteria, that genetic information from one bacteria can be transferred to another bacteria, giving that other bacteria the same characteristics of the first bacteria. And that's known as transformation. In other words, the R strain was transformed into S strain. Griffith's transformation. So now we know that, and so that was a huge discovery in itself. But what this still leaves as a question is what? What is that genetic material that was transferred? That's what this question mark is all about. We still don't know what. We know that genetic information is transferred from one bacteria to the other, but we don't know what that genetic material is. So again, I just carried this over from the previous slide to remind us what we're after. We're trying to figure out what this genetic material is. And at that time, there were two camps, as we say, in other words, groups of scientists who believe one way or the other. There was one camp that thought that it was DNA, and there was another camp that thought that it was protein, because both DNA and protein have the characteristics that allow for having information, containing information, just like the letters in words. There's a sequence to the letters in words, and that spells out information. Well, there's a sequence to nucleotides in DNA, and that can spell out information. There's a sequence of amino acids in protein, and that can spell out information. And actually, it kind of makes more sense that protein might have been the genetic material, because there are 20 different amino acids. So there's like a 20-letter alphabet, very similar to our 26-letter alphabet that we use to spell out the English language. DNA only has 4 letters. So it was in that way, if you look at it like that, it's less likely that DNA is the genetic material. Another scientist, 1940, Oswald Avery, did an experiment where, and he followed up on Griffith's transformation. He used an extract of the heat-killed S-strain bacteria. So he's basically repeating Griffith's experiment, but he's adding another step to it to try to figure out what the material is, what the genetic material is that's being transferred between bacterial cells. So what he added were hydrolytic enzymes that break down organic biological compounds. And there are specific hydrolytic enzymes for specific organic biological compounds. So he added enzymes that are known as proteases that break down proteins. He added lipases that break down lipids. What I'm calling carbases, there really is no such thing as a carbase, but I'm calling them that because they break down carbohydrates. And nucleases break down nucleic acids. Notice they all end in ACE, A-S-E. That tells you that they are enzymes. And the names also tell you what they do, what they break down in this case. So when he added all these hydrolytic enzymes to Griffith's transformation experiment, transformation did not occur. And that's no surprise, because everything that could have been the genetic material and transferred from one bacteria to another was broken down. All the proteins are broken down, the lipids are broken down, carbohydrates are broken down, and nucleic acids are broken down. So there's nothing left to transfer. But in his second experiment, he left out the nuclease. And, well, actually, no, he used a specific nuclease that was DNase. In other words, it's specifically he left out the enzyme that specifically broke down DNase. And he found that transformation did occur. So basically, he destroyed everything else that it could possibly be except for DNA, and transformation occurred. And so from that, he concluded that, like it says here, it's DNA, that the genetic material is DNA. But the protein camp still thought that it was protein. You know, being good, critical, skeptical scientists, they felt that there was still a strong possibility that proteins could be the genetic material. So we need one final experiment to finally show that DNA is the genetic material. And this was conducted by two scientists named Hershey and Chase. And what they used for their experiment were bacteriophages. Bacteriophages are viruses that infect bacteria. So normally you think of viruses infecting people and infecting animals, and there are viruses that infect plants, and pretty much every living thing on the planet, there are viruses that will infect them. So these are viruses that actually infect bacteria. And what's perfect about them for this experimentation is that they are made of nothing but the two compounds that were suspected as being the genetic material. In other words, they're made of nothing but protein and DNA. The outside of the virus, basically the whole body, I guess you could say, of the virus is made up of protein. It's known as the capsid. The whole outside of the virus, all these different components that are labeled here, is known as the capsid. And then inside this icosahedral head here of the virus, there's DNA. So they're nothing but protein and DNA. So it turned out that it was the perfect experimental subject to use to try to figure out what the genetic material is. Viruses are what we call obligate intracellular parasites. They're non-living, considered by most biologists to be non-living. Obligate means they have to. Intracellular means inside cells. And parasite, I think you know what that means. So they have to parasitize cells, and what they do is they get their genetic material into the cell. So the first thing they do is they attach to the cell. They inject their genetic material. And again, remember, that's what we are trying to find out what it is here. What is that genetic material? They knew at that time that the genetic material was injected, that the virus injected its genetic material into the bacterial cell, but they didn't know what that genetic material was. The virus then, the genetic material is expressed, making components of the virus, the different proteins, and copies of the genetic material that go into reforming new viruses. So the viruses are replicating in this way. Notice that it's replication and not reproduction. We usually use the term reproduction for living things. We're using replication here because viruses are considered non-living. But anyway, the viruses are then, once they replicate, the cell breaks open, releasing all those replicated viruses that can go on to infect other bacterial cells. So that's how viruses operate in general. They hijack living cells to make copies of themselves and destroy the cell in the process. And that's what makes you sick when you're infected with a virus. It's the destruction of your cells that makes you sick. So because they knew how viruses worked and they knew what viruses are made out of, they were able to chemically tag a tracer molecule, tracer isotopes onto either the protein or the DNA of the virus. And then they allowed the virus to infect the cells, and then they looked to see where the radioactivity was, to try to show what was being injected into the bacterial cells. So in this first experiment, they did 2 experiments. In the first experiment, they used S35, which is sulfur 35, and there is no sulfur in DNA. There is sulfur in protein. So it turned out that the protein was the only thing that was tagged with this radioactive sulfur 35 isotope. Then they allowed the virus to infect the cell, and then they shook the viruses off and checked to see if there was any radioactivity inside the cell. If there was radioactivity inside the cell, then that would suggest that the genetic material was protein, because the only thing that was tagged here with radioactivity was protein. But they didn't find any radioactivity inside the cell. All the radioactivity was outside the cell. So that refuted the hypothesis that protein is the genetic material. They then repeated the experiment, but this time they used phosphorus 32. And it turns out that there's phosphorus in DNA. There's a lot of phosphorus in DNA, actually. You'll learn that in this unit. But there's not much phosphorus or no phosphorus in protein. So that selectively tagged the DNA in the viruses. And they did the same thing. They allowed the virus to infect the cells, and then they checked to see where the radioactivity was. And they found that the radioactivity was not outside the cells, it was inside the cells. So that showed once and for all that DNA is what was injected into the cells by viruses. So when they inject their genetic material, what they're injecting is their DNA. So the bottom line is that these results indicated that the viral DNA entered the host cells, supporting the hypothesis that DNA is the genetic material. And this really is revered as the final experiment to show this. And if you clicked on that big word that says click in the upper right-hand corner of that slide, this is the video that it takes you to. So I just want to go ahead and play this for you so you don't have to worry about trying to find it yourself.
In the Hershey-Chase experiment, bacterial viruses called phage.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Don't understand how they did this experiment, you'll want to go back and watch this video again or go through the notes that I just explained. So where we are now is this. We just finished looking at the history of the discovery that DNA is the genetic material, but we still need to know what the structure of DNA is so that we know how it does what it does. And that's what we're going to be doing in the next section.